Good afternoon, everyone. Welcome to this session of the Chpc conference. Our first speaker today is Professor Marlo Möller. She is professor in the molecular biology and human genetics at the Stellenbosch University. Thank you, Professor. Go ahead.
Thank you, Ines. Is that all good? Great. So good afternoon, everyone, and thank you to the organizers for inviting me to give this talk. I'm very excited to tell you a bit about the work we're doing in our group. So the title of my talk is The Infectious Disease Susceptibility in the Context of African Genetic Diversity. And I want to start off with a number of acknowledgements first. This work would not be possible without our collaborators and a couple of institutions and funders. And I'm not always going to name all the names today. There's too many. But please know that I'm very thankful for your contributions. So I am part of the TB host genetics group, and we are specifically interested in finding out why some people go on to develop TB while others don't. And we focus on the host genetics that contribute to this. So we do association studies, we do functional analysis to verify our findings, we focus on extreme phenotypes of disease, and we bring in the unique population genetics of South African groups in our analyses. So today I'm going to talk a bit about the background of infectious diseases, specifically comparing TB and COVID-19. And then I'll discuss the evidence for genetic influence in infectious diseases. I'll address a few basic concepts in genetics. I'll tell you guys why we need the Chpc. Then I'll move on to genetic diversity in South Africa and how we've applied it in our studies of genetic susceptibility to TB. And then I'll talk about how we are using the lessons we've learned in TB to apply to our study of COVID-19 host genetics. So if you compare TB versus COVID-19, TB is an ancient disease. It's been around for a very long time. It's obvious in mummies from Egypt, while COVID-19 is a very new disease that only originated in 2019. For TB, it took 39 years after the discovery of the pathogen before we had our first vaccine. And it was, of course, very different times. And now we've got newer technologies that made it possible to develop a vaccine for COVID-19 much faster. Another problem with the TB vaccine is that it's only effective in children, and we only have this one vaccine, whereas for COVID, we already had 18 by June 2021. For TB, there's only 14 vaccine candidates at present, and for COVID, we have 109 already. And you'll see why this is. It's just that there's much more money being spent on COVID-19 at this time. Even so, the death rate is comparable between these diseases if you look at the annual death rates. So TB is caused by a bacterium and COVID is caused by a virus. So it's two different pathways that will definitely be involved with this. So bacteria, you'll see there's a few things listed, the differences between bacteria and viruses. Bacteria tend to be bigger, they've got a different structure around them, and they also have a different hereditary material. They have different survival strategies. And we consider bacteria as being living organisms, whereas viruses are considered being non-living. So this is the estimated TB incidence rates in 2020. And you can see that it's mostly the developing world that is affected by TB, whereas there's a lower incidence rate in the developed world. And this is probably because of better socioeconomic circumstances in these regions. South Africa especially has a high TB incidence, one of the highest in the world. And for COVID-19, the picture is a bit different. There we're seeing a higher incidence in the developed world compared to the developing world. And this can be for several reasons. It could be due to reporting that's not as great in these regions, but it could also be because the developing world has a younger population. And we know that COVID affects the elderly more severely. But I like to think that there might be other reasons, and we are hypothesizing that genetics could be involved here. So after exposure to tuberculosis, the causative bacterium in TB, you can get several outcomes. When you're exposed, the infection can be eliminated by the innate immune response or by the acquired immune response. In other words, you will have a memory of having encountered TB. In most people, we see a latent infection established, which is so-called latent infection, where they never have symptoms of the disease, they will never test positive. And in South Africa, we don't even give preventative therapy because it's so common. Then you also get subclinical Tb disease. These individuals will test intermittently positive for TB, and they'll have mild or no symptoms, and they sometimes not even detected. And then you get active Tb disease, where we usually have a positive test, they're infectious, they have mild to severe symptoms, and they also need definite treatment. So if you compare this to the outcomes of SARS-CoV-2 exposure, you can see it's very similar, as we're all aware now. We do see some individuals that seems to be resistant to infection, and they're really interesting for genetic studies. And then on the other side of the spectrum, we see younger people who get very severe disease, even when they don't have comorbidities. And then we see all the other outcomes in between, so mild or asymptomatic responses. In some children, we also see a very rare syndrome called multisystem inflammatory syndrome related to COVID, MIS-C. They present usually 2 weeks after they had SARS-CoV-2 infection, and they're also very interesting in terms of genetic studies. So TB is a complex disease. Just because you're infected doesn't mean you'll get sick. You need the perfect environment and also a susceptible host to go on to get active disease. This is also true for COVID-19, and as you might know by now that several comorbidities can contribute to it, but we also hypothesize that the genes that play a role in the immune system contribute to outcome. So what's the evidence for genetic influence in infectious diseases? Well, when a naive population is first exposed to an organism, a pathogenic organism, they will be really susceptible. And in the case of TB, there's this example of the First Nations in the Americas. When they were first exposed to the TB bacterium in the 1880s, the death rate was 10% per annum. After 40 years, this dropped to 0.2% per annum. So that's an indication that there was a very strong selection against the susceptibility genes. And I guess we can equate that to what we're seeing now with the COVID pandemic. If the death rate will fall like this after several years remains to be seen, because we've got other interventions, of course, but it's definitely a possibility. Then another observation is the wide range of responses to infections that we see, and as I discussed. And for TB, there's this very horrible example where they injected babies with a live TB strain by accident in 1926 in Germany. So although all the babies received the same injection and the same amount of the live TB, only 47 showed no evidence of disease and 77 died. We also see evidence for genetic influence in infectious diseases in twins. So identical twins are more likely to both have disease compared to dizygous twins in TB. And there has been some anecdotal evidence that this is also the case for COVID-19. And one of the best studies is a classic of evidence for genetic susceptibility to infectious diseases is this classic epidemiological study on the premature death of adoptees in Denmark. So if the parent died due to an infectious disease, their child who was adopted and raised in another environment was also more likely to die of an infectious disease. And the risk for that was greater than for cancer or for cardiovascular disease. We also have examples of monogenic disorders that present as an extreme susceptibility to an infectious disease. For TB, we have what we call Mendelian susceptibility to mycobacterial disease. And these individuals are really susceptible to even normal avirulent mycobacteria and salmonella. And individuals usually carry mutations in the IL-12, IL-23 or interferon gamma axis. We also have animal models of disease. And in the case of TB, the mouse model helped us find the first TB susceptibility gene. So what is DNA? And anyone who has watched an episode of CSI should be able to tell me this. So DNA is everywhere in your body. It's in your saliva, in your blood, in your skin, and even in your hair. And you can think of your DNA as a book. Every page in your DNA book is called a gene. And your genes give the instructions to make you. Now, DNA is transferred from parents to their children. And this is why you look like your parents. Your parents inherited their DNA from their parents, who inherited it from their parents, and so forth. And your children will inherit 50% of your DNA. So what we do in studies of disease is look at genetic variation. So the percentage of DNA that's shared among all people is the same between 99.9%. So it doesn't matter where you're from. We are also 65% similar to the chicken, 84% similar to the dog, 85% of our DNA is shared with a mouse, and then we share 98% of our DNA with a pig. And I always like to joke and say, but this is why some people drive like they do. So we look at single nucleotide polymorphisms that forms part of our DNA, and our DNA consists of 4 chemical bases. So these single nucleotide polymorphisms is a single change in a DNA base. And these SNPs generate biological variation between people, and this causing differences in the proteins that we create from these genes. And they can influence things like your susceptibility to disease, your response to medication, but also some of them we don't have any evidence that they do anything. So classical genetic study designs, one is the family linkage study. There you look at many members from the same family. And by looking at shared regions of DNA between them, it can help you track down the mutation that contributes to disease. And then you go on to develop your biological hypothesis. For candidate gene associations, here you go in a different direction. You first come up with your biological hypothesis, then you go into the genome and find the region where these genes are located. And then you compare cases and controls for this region to find the variant of interest. And recently, or not so recently anymore, we are now able to look genome-wide. So that means we can compare cases and controls for millions of markers across the genome, see if there's a difference in frequency between the SNPs, and then identify common variants that may be associated with disease. And this helps us to come up with a biological hypothesis of how these variants contribute to disease. So for genome-wide association studies, we use microarrays, or as we affectionately call them, chips, to genotype people. And this is not similar to this chip in your cell phone that can allow you to be tracked. But it allows us to genotype large number of people over millions of SNPs. And recently, we're also able to do human whole genome sequencing in South Africa with the SAMRC Genomics Center. And this allows us to look at the whole human genome across all genetic variations. It's of course a bit more expensive than the microarrays, and you tend to not do it for such large sample sizes. But I think once we have the budget to do this, we definitely will. So why do we need the CHPC? The files that we receive from both microarray genotyping and the sequencing platforms, it's really large because it's across many individuals. And this is too big and difficult for a normal computer to handle. And that's why we're so thankful for the structure offered by the CHPC. So just to show you guys that this is my ancestry, my genetic diversity. So the phenotype doesn't always correlate to the genotype. And I've got ancestry from across the world. I skipped a slide by mistake. So South Africa is a hotspot for genetic diversity because we have ancestors from all over the world, but we also have the unique click-speaking hunter-gatherer groups here, and they've got lots of genetic diversity. So this is why Africa and South Africa is so popular for genetic studies. And as I pointed out, this is my ancestry that reflects what we would expect to see of someone from South Africa. So our group has been doing TB host genetic studies in South Africa since the 1990s. We've established this large biobank, and most of our work is centered around this. And we've got TB cases, so that's actually someone who had an active TB case or previous TB. And then we include controls. So those individuals are from the same high incidence communities, but they've got no evidence of clinical disease, and they're unrelated to our TB cases. And we've done the largest TB GWAS meta-analysis to date. This was done by Heiko Schutz with Vivek Narenbai. And Vivek managed to do 12 cohorts of TB cases together. That's almost 15,000 TB cases and 17,000 controls from different continents. And one significant association was identified. Now, this is fairly typical for TB association studies, where they don't seem to replicate across different populations, or where they are not found again. And we think this is because of regional differences between human populations. And our work has centered on this, and we've investigated genetic ancestry in many South African groups and found some stretches of DNA that we can use to trace genetic variants that are associated with TB. And as part of this, we've developed some genetic tools and approaches to help work with these complex South African populations, because the genetic ancestry is so admixed and much more complex than what we see in other parts of the world. We've also looked at host pathogen coevolution for TB. So we can see that the Mycobacterium tuberculosis strains has co-evolved with the host populations in the region where they originated. And some of our studies have shown that different TB strains are more likely to infect people with specific host genetic components. And this was published earlier this year, where we looked at a cohort from Ghana and South Africa and could find these associations. And then Caitlin Uren also did a very nice review on this topic, explaining how this works. Then we also study extreme forms of TB. So we get very susceptible individuals, as I described earlier, and some individuals with primary immune deficiencies are more likely also to get TB and have multiple disease episodes. And then we also look at individuals with no evidence of TB infection, or the so-called TB resistors. And we're working to find out how these genes contribute to these phenotypes. So what have we learned from TB genetic susceptibility? Well, we know that ancestry matters in genetic investigations of TB because they can affect our case control association studies and skew them, but it's also a powerful tool to help us track down genetic variants associated with disease. We know that ancestry can help determine our susceptibility to specific TB strains, and it can also affect predisposition to develop extreme forms of TB. So for COVID host genetics, there's been quite some goodwill across the world, and some large consortiums have been established. So the one that we are part of is the COVID-19 Host Genetics Initiative. They published their first paper in this year and identified 13 genomic loci associated with severe COVID. Unfortunately, they had no samples from Southern Africa yet, but we are working on this meta-analysis with them. And then there's another big initiative, the COVID Human Genetic Effort. They are more focused on inborn errors of immunity, so individuals with specific mutations. And for 10% of critical COVID-19 cases, they found mutations in the type 1 interferons. And they also found autoantibodies against the type 1 interferons for some of the other patients. So where our study comes in is to contribute Southern African genetic data, and we're trying to find out why South Africans specifically get very sick. We collect the DNA by scraping the inside of the cheek using a buccal swab, and the participant can do this himself. So we ship the kits to them and they just return it to us. And it's painless and nothing like a COVID test at all. So our progress to date, we've whole genome sequenced 71 individuals with the SAMRC Genomics Center. This includes study participants that were hospitalized with disease, but also children with MSC and controls. And the data was deposited online. So it's available to other researchers with questions on this topic. And this work is part of the master's degree from Crystal Stale, who's been using the CHPC extensively for this work. So Crystal has already found some genes known to be associated with the immune system pathways, and these are specifically based in the adaptive immune system, in the innate immune system, and also in some cytokine signaling pathways. We have also banked samples from more than 600 hospitalized participants with COVID-19, and we've recruited more than 400 individuals from the general public who tested both positive and negative, because remember, we need controls, for SARS-CoV-2. We're also collecting information on their vaccine status. And these individuals will be genotyped on a specific array that was made by Thermo Fisher to look at the host genetics of SARS-CoV-2. So why is our study important? Well, for South Africans, it's important that Africans should be included in these studies so that when there's new treatments coming up, that we can take part in these applications and not be excluded. It might also be the case that novel variants are associated with COVID-19 in Africa, and that can be with susceptibility or resistance. For the COVID-19 pandemic, it's important that we understand the genetic causes behind the severe and critical COVID-19 disease, and it might also help us to better understand the disease outcomes of COVID-19. Importantly, if we can identify those individuals that are genetically susceptible, we can come up with revised vaccination and treatment strategies. And then for the MIS-C study, this is also the first genetic study of MIS-C in South Africa or in Africa, and it can help us to figure out why there's this difference between children and adults, especially for this phenotype, because you get the same phenotype in adults, but then it's just called MIS-A. So in conclusion, we hypothesized that ancestry will matter also for genetic investigations of COVID-19, and it's important that these studies should be representative of the world's genetic diversity, in other words, including samples from Africa as well. We think that there might be some contributions that some individuals can be susceptible to specific variants of COVID-19, similar to for the NTB strains. And it's also possible that the ancestry could affect the predisposition to extreme forms of COVID-19, such as MIS-C. So I would just like to thank everyone who has listened to the talk today. And I'm just promoting a few cases close to my heart, so please feel free to visit their websites. Thank you very much.
Thank you, Professor, for the very interesting talk. If there are any questions, please type it on the question mark on your on the call, and then it will be relayed to me via the Zoom to come in here. So far, I haven't seen any questions. I have a very stupid question in a way: is when will this epidemic be over? The pandemic?
To be honest, in my opinion, I don't think it's going to go away, Ines. I think we missed that window. If people had been more compliant and gone for the vaccine when we had access to it, if it was more equitable, I think we might have stood a chance. But I think now that we're seeing so many variants that it's probably going to be around for a while still in different guises. I think we're already seeing the new variant. I'm hoping that the predictions are true and that it's going to cause less severe disease, so that it will become like the common cold in future, but we'll have to wait and see. And I think one thing about COVID-19 is, and it's the same with TB, we always have this dogma. If you think back to the start of the COVID-19 epidemic, we thought that the virus wasn't mutating, right? We thought it would stay the same. And it's certainly proven us wrong in that. So I think any prediction is just that, and we're learning as we go along.
Well, I'm impressed to be part of the small contribution we can make to the research, to the science, and let's hope that a breakthrough comes soon.
Thank you, Ines.
Okay, we don't have any other questions. Okay, there is one coming. Reading, thanks for the presentation. Do you believe that since we have this unique sequence in South Africa, that moving towards a precision medicine approach of developing drugs is the way to go, rather than having everyone treated with one centralized vaccine? And this is from Krishna Govinder.
I think it would be very nice if we had that option. I think at the moment, because of the intensity of the pandemic, it might not be possible to have a personalized approach. But that's definitely what we're working towards as geneticists. In one of my other studies, we had this comment from a clinician saying, oh, we'll never have personalized medicine in Africa. It's too expensive. It's not possible to roll it out here. Well, I beg to differ, because I think if you look at the cost implications of not performing personalized medicine in Africa, that will be much higher in the long term. But that's my personal belief, and of course, we have to convince stakeholders of that still.
We've got another question from Quinn. He says, thank you, Marlo, fascinating work. To follow on from Krishna's question, how far away do you think we are from personalized medicine based on genomics, which could be especially impactful in South Africa's highly genodiverse population?
So we've already moved towards that in South Africa. And in fact, for some of our other work, we have identified a mutation in South African individuals that's associated with a phenotype that's pretty common. And it's it's already been rolled out with the NHLS, so the test is available to people. So we actually do have personalized medicine rolled out for rare diseases especially, and I think we'll move that way more and more and more. So we've got Dr. Shahida Musa in our division together with Prof. Mike Urban, and they're really working with the rare diseases. So it's already a reality. Through those clinicians, we also have contact with Invitae. It's a company that's based overseas, and they do whole exome sequencing, targeted sequencing, and genome sequencing to help with diagnosis. So if you have a rare disorder, I think there are options.
Thank you. We've got another question. It says, good day, Prof. Are there similar studies globally? If yes, waiting for this is the last part. But are there similar studies to the TB one, I suppose, globally?
Yes.
And
so
what makes this one unique?
So with the TB studies, I think what makes our study unique is that we've got this unique genetic diversity. We're finding different variants that's associated with the disease. If you remember my slide that I had up, where we did the meta-analysis between 14 different nations, we only found one genetic variant in common. So that tells me that the human and the pathogen probably evolved together, and we're seeing unique associations. So no, we're definitely not the only group working on this, and the same for COVID. There's been really a huge push from everyone to come together and do genetic studies for this as well. And I just named 2 of the consortiums contributing to it at the moment. There's more.
Thank you. Okay, I think this is the wrap, and thank you very much for your presentation. And our next speaker will be Professor John Mack.
Hello, yes, I'll just share my screen at this point.
Okay.
Okay, so can you see the screen okay and can you hear me? In which case I will get going. So the title of my presentation is The Rational Design of Tin-4 Porphyrins for Photodynamic Therapy, Further Progress and Future Perspectives. I've carried out this research at the Institute for Nanotechnology Innovation at Rhodes University. I collaborate very closely with Professor Tebelo Nyakong, and there's also been a collaboration with Brazil that was involved in this project over the last couple of years. So, what's the background to this? We're looking for molecular dyes that can serve as photosensitizer dyes to generate singlet oxygen and other reactive oxygen species, and so that can be used to destroy cancer tumours essentially. Idea is that the molecular dye is introduced intravenously. We're hoping that it localizes in the target tissue. We'll get back to that question later. We use laser light, usually in the red region, near infrared, to generate the singlet oxygen and destroy the cancer cells so that the patient can recover without a need for chemotherapy, without a need for surgery. This is basically a much less invasive type of treatment, and anywhere up to about 10 millimeters from the skin surface, there's some scope for using this approach. So, basically talked about how we absorb a photon of light with a molecular dye. We generate a triplet state, and either via energy transfer to form singlet oxygen or electron transfer to form oxygen radical species, we can form species that can destroy the tumours. And basically, going back to about 1975 or so, the first molecular dye that was used in this context was referred to as photophyrin, and it was a porphyrin oligomer. And the big issue that we have with porphyrin dyes for this application is that they only absorb very weakly at the red end of the visible region. I'll come back to why that's important in a moment. And basically, if we're going to use laser light within human tissue, there's a window into which there's substantial transparency of human tissue. If we go to the blue end of the visible region, 400 nanometers up to about 600 nanometers, we have an issue with hemoglobin. And if we go beyond about 850 nanometers, beyond 1000 nanometers into near infrared, there's an issue with water potentially absorbing the incident light. So there's a window between about 650 and 850 nanometers where we really want to be for this application. And from an African standpoint, melanin is a major issue, and we really want to be as far red as possible in the African context, out to about 800, 850 nanometers. So the initial dyes that were used for this weren't so promising from that sort of standpoint. And where have I used the resources of CHPC? Essentially to look at, with geometry optimizations and with TD-DFT calculations, looking at how the spectroscopic properties of the porphyrin ring structure can be modified by carrying out various different structural modifications, and by looking at trends in the energies of the frontier orbitals, we can look at how we can narrow the homo-lumo gap and shift the spectral bands further into the red to lower energy and out into the near infrared. And by modifying the structures of the porphyrin molecular dye, we can potentially come up with new photosensitizer dyes that are more suitable for this application. So basically, if porphyrin is our starting point, the three major routes that we've been looking at Rhodes University, at the INI at Rhodes University, is to modify porphyrin by reducing a peripheral bond to form a chlorine. This is related to chlorophyll, not exactly the same as chlorophyll, but similar. And for similar reasons to chlorophyll, you can shift the spectral bands further to the red. We've also looked at chirols, where we've got a direct bond between two pyrrole rings, and to N-confused porphyrins, where one of the pyrrole rings that makes up the porphyrin ring is flipped over so that the nitrogen atom is on the periphery of the ligand. That's referred to as an N-confused porphyrin. So basically we've used CHP resources to identify the structural modifications that are most likely to give us dyes that have enhanced properties in terms of the spectroscopic region where the dye absorbs. And there's also the question of the stability of the dye. If we, I'll go back a slide or two, if we stabilize the HOMO level, the highest occupied molecular orbital, if it's stabilized, the molecule is not going to be as prone to oxidation and is likely to be more stable in the context of the application. So it's many things going on here that we need to consider. And starting about 2019, what we were aiming for was dyes that are as easy as possible to synthesize and to scale up from an industrial standpoint. And our starting point was tin-4 porphyrin complexes. And the advantage here is that because we've got this axial ligation, we can limit the amount of aggregation of the dye. The dye is prone to pi-pi stack. That's not good from a photophysical standpoint, and from single oxygen generation. And the other advantage of tin as a central metal here is that it helps to enhance the formation of the triplet state by intersystem crossing. So that was our starting point. Our initial interest with porphyrins was that if we changed the ring system that's introduced at these bridging carbons, we found that we could narrow the homolumal gap and redshift the main spectral band, making it potentially more suitable for use in the application. We were able to move the porphyrin band from about 600 to 620, and when we carried out cell studies and had IC50 values with cancer cells and with laser light, we found that this structural modification was significantly enhancing the rate of singlet oxygen formation and the concentration required to get 50% elimination of the cancer cells, that's what an IC50 is referring to, was significantly lower when we made the structural modification with these thiophene rings. So that was our starting point, was these porphyrin rings. And moving on from there, we looked at other structural modifications that could be carried out to have a greater redshift of the spectral band into the region where we need to be for transparency within human tissue. So moving on to corals, we again use the same thiophene ring system, and with a direct carbon-carbon bond we were able to shift further into the red where we really need to be from a transparency of human tissue standpoint. And again we were using molecular orbital calculations, TD-DFT, on the Chpc system to figure out which particular set of structural modifications was going to give us the most suitable properties from this standpoint. And again we were getting favourable results in terms of IC50 in cell studies and making progress. So moving on from there, there's other possibilities that we looked at. So another possibility that we've looked at, which we identified by molecular modelling as a strong possibility for enhanced properties, was the introduction of bromine atoms to the 8 peripheral positions of the porphyrin ligand. Again, we're using tin for the reasons that I described previously, and essentially we get a red shift of the main spectral band into the 650 to 700 nanometer region, and now we're really in business where 650 to 850 is the optimal range that we really want to be in. So again we used molecular modelling, and that made us confident beforehand, before we even got into the synthesis and carried out all the cell studies, that this was an approach that we wanted to pursue. So using molecular modelling to rationally select the dyes that we want to carry forward to synthesis and into cell studies. So again, with this octobromination approach, we were getting enhanced properties when we moved on to cell studies, and packed here is similar to photodynamic therapy that I described at the beginning, but with bacteria. So moving on, another structural modification that we've looked at is chlorines, where we have a reduced peripheral bond. Again, we've gone with the thiophenes, which we've found helps to enhance the redshift of the spectral band. It also helps to enhance intersystem crossing to the triplet manifold, which helps us to generate the singlet oxygen. And with this modification we're moving from about 600 to 664 nanometers. And we've got a whole series of free base chlorines at this point, and depending upon which ring system we used, we had different amounts of red shifting of the spectral band that we were interested in. So again, further modification of the rings on the periphery can make a significant difference to how far red we can go with the spectral band. And we were obtaining suitable singlet oxygen quantum yields. What these ratios refer to here is that 0.6 means that 60% of the incident photons are forming a singlet oxygen molecule, which can help to destroy the cancer cells. And again, with these chlorines, we were getting a significant improvement when we moved into cell studies with substantially lower IC50 values, the concentration that you need to decrease the cell viability to 50%. So chlorines is looking like it's possibly the way to go. Again, we also looked at an antibacteria approach here. We're looking at Staphylococcus aureus and E. coli as typical bacteria. One of the problems from a bacteria standpoint is hospital superbugs generate resistance to antibiotics, so there's a need for alternative treatments. So the photosensitizer dyes can also be used in that context as well. If you've got some sort of staph infection on the surface of the skin, one possibility is to use what's referred to as photodynamic antimicrobial chemotherapy to use singlet oxygen to destroy the bacteria. So basically we moved on from there. We also looked at what happened with tin complexes of these chlorines and how that modifies the energies of the frontier molecular orbitals, and how that helps us to redshift the spectral band and also to intensify the spectral band in some instances. We'll look at trends in these frontier orbitals, and that can help us to rationally select the dyes that we want to carry forward into experimental work. And again, when we were looking at the tin complexes of these chlorines, we were obtaining substantially enhanced IC50 values in cell studies where we use molecular dye and laser light to destroy the cancer cells. So moving on, another type of porphyrin that we have looked at, let me just have a look in the chat. Okay, I'm being told it's about 5 minutes to go, no problem there. I'll definitely be finished in time. So with N-confused porphyrins, this was really the one we're most excited about from an African standpoint, because the manner in which we modify the frontier orbitals helps us to shift the spectral band out towards 800 nanometers, where we really want to be with these photosensitizer dyes. And again, we carried out cell studies. We obtained very promising results, not so much at 780 but more at 660, but we're looking at ways to further enhance the structure of these dyes to get the set of properties that we really want. Here we've further modified the N-confused porphyrin. We're able to absorb out to about 850 nanometers with a very broad and intense absorption band this time. And again, we were obtaining reasonably good IC50 values. We still need to do more structural modifications in this type of dye to really be in business, but we're identifying sort of the platform that we want to build on to get to the dyes that can be carried through to clinical trials. Something that's easy to synthesize, easy to scale up, and gives us all the properties that we want. So moving beyond that, we're also looking at conjugating the dyes to gold and silver nanoparticles. That helps in terms of the transport and selective accumulation within cancer cells, cancer tumours, and I've been working closely with researchers in Brazil in this context. Unfortunately, we've really not been able to do exchanges because of the pandemic, unfortunately, over the last couple of years, but we're hoping to get BRICS funding for the next 3 years. We've certainly applied. We'll see what happens. Recently, we've had publications looking at what happens when we can conjugate the dyes. These are chiral dyes, conjugate them to gold nanoparticles. And our initial results in that context have been quite promising. So, CHPC resources, we can use very large memory allocations for the TDDFT calculations compared to what is possible on a desktop, and that makes a huge difference in calculation time. It makes a huge difference on the size of the molecule and the complexity of the molecule that we can easily calculate the properties for. So although it's not the most high level from a computing standpoint use of your system, we still find CHPC extremely useful to be able to push these calculations to the outer limit of what's doable, essentially. So we use CHPC to identify rationally the dyes that we want to carry forward, and the goal is to come up with the best possible porphyrin analogues from the point of view of ease of synthesis and scale up, and properties in terms of IC50, enhancing their solubility, lipophilicity, so that we have dyes that we hope in the not too distant future can be carried forward further into clinical trials. We have collaborators in China who can do that sort of thing if we come up with the right set of dyes, and also our friends in Brazil that I mentioned as well. So to finish up, I should express my thanks to Rhodes University, Professor Tibala Mearkong, for giving me the opportunity to carry out this type of research. Yeah, I think you can tell from my accent that I moved to South Africa specifically to work in an environment where I could carry out the type of research that I was trained for and that I really wanted to take through as far as possible towards something practical and moving away from sort of blue sky theoretical research towards something that's actually useful. Thank the funders, Rhodes University, NRF, for an international collaboration, and I said CSII here, but that's essentially CHPC. Okay, so I'll stop the share at this point, and I will allow Ines to step in.
Okay, thank you very much, Professor Mack, for the very interesting talk. I would like to remind people that they must use the questions tab on the conference site. It's got a question mark on it, and if you can type your questions in there. I have a question for Prof. Mack, and that is, are there any of these products already in production, and the kind of research that you do, in what time scale would a practical therapy come out in terms based on your research?
There's 10-4 complexes moderately similar to what I'm talking about that are in advanced clinical trials. Right now, I think, if I was being honest, it's still somewhat distant that any of our dyes would get there, but I think you can see that we're building up the complexity of the dyes as we go along. So, maybe 5 years, 10 years, hopefully we'll get there.
We have a question from Dr. Krishna Govinder, and he says, thank you for the presentation, John. Have you considered having a look at what effect these dyes might have on healthy cells en route to the cancer cells?
Indeed, yes. I skipped over that point, but I mean, one of the things we do is we ascertain that there's no dark toxicity. So it's very important that the dyes don't have an effect on the normal healthy cells in the dark. So one of the steps we do before we do the experiments with light, the photocytotoxicity experiments, is dark toxicity. We just leave it in the cells to ensure that the cell viability is not compromised in the dark. And it's only that when we shine the laser light and we generate the singlet oxygen species that we start to destroy the cells.
I think you've answered Quinn's question essentially, which he said, thank you, John, very impressive work. I noticed that you presented many of the performance graphs as functions of dosage. Is there significant toxicity associated with these materials?
Essentially, we're hoping that you need to get up to about 50 micromolar or 100 micromolar before we're seeing a dark toxicity effect, and that we can get down to about 1 micromolar as the dosage for the photocytotoxicity. And then at that point, there's not a huge concern about the effect in healthy cells, but obviously you need to get into clinical trials, and there's many steps to go before you get into clinical trials with humans, like it would be with mice initially, and you go through animal testing to clinical trials with humans, and then it gets onto the market. So it's still a long way to go.
Okay, if there are no other questions, I'd like to thank you very much again for the presentation. We've got another 3 minutes or so maybe if another question comes through.
And
Otherwise,
I guess I should, yeah.
Otherwise,
just
this will be
gonna
the end of the session. Mm-hmm.
Yep. I was just going to step in and say thank you for the organisers for allowing me the opportunity to give the presentation.
Okay, thank you. I think we will close the session, yeah. Thank you very much, Professor Mack.
And thanks for being the organiser. Thank you.
All right. Thank you.